we were going through the structural features of the second most important fibrous protein, that is collagen. And we have already gone through the structural features of the fibrous protein keratin. So collagen is a kind of protein that is well found in connective tissue, the cornea of eye, then tendons, the organic matrix of bone, then cartilage, then ligaments, etc. It promotes structural flexibility and again is a kind of, again has several, that is a essential structural role to perform. It makes up the structure of the organic matrix of bone, it makes up the structure of cartilage, tendons, cornea of eye, etc. So collagen is the element, the secondary structural element of the tertiary structure or the three-dimensional structure of collagen is actually an alpha helix, but the alpha helix of collagen is unique in itself because it has a unique structure quite different from that of the regular pattern of alpha helix. That the alpha helix which is found in collagen, instead of being right-handed, is left-handed, which forms the individual secondary structural element found in collagen. It is left-handed and consists of 3 amino acid residues per turn. I hope, think you all remember that alpha helix, the twist of alpha helix found in proteins is usually right-handed, and alpha helix consists of 3.6 amino acid residues per turn. So an exceptionally different or an exceptional form of alpha helix is present in collagen, which consists of 3 amino acid residues and is left-handed. And moreover, the alpha helices of, so collagen has a super-secondary structure, or the super-secondary domains or super-secondary motifs of collagen are made up of several repeating elements of the secondary structural element called alpha helix. That is, a number of alpha helices are again twisted and coiled to form a super twisted coiled coil called the collagen. So it's made up of number of alpha helices which are unique in itself, and every alpha helix is made up of 3 amino acid residues and is left-handed. Now if we consider the super secondary structure of collagen is, that is the tertiary structure, the super secondary motif structure of collagen, or the 3 degree, that is tertiary structure of collagen, we'll come across the fact that it is actually made up of, or it consists of 3 different chains called alpha chains, or polypeptide chains called alpha chains, which are quite different from that of alpha helix, which are not to be confused with alpha helix. And these alpha chains are actually twisted and coiled around each other to form a super twisted coiled structure of collagen. So collagen consists of 3 different alpha segments or 3 different polypeptide chains called alpha chains, which are not to be confused with alpha helix and are quite different from that of alpha helix, and they are wrapped around each other to form a coiled super twisted helical structure called collagen. So collagen now has, invertebrate collagen has a unique sequence of amino acid residues, and all vertebrate collagens have 3 amino acids in common. That is, almost all of the vertebrate collagens consists of or have in their structures are made up of about 35% of glycine residues, then about 21% of alanine residues, about 11% of proline, 4-hydroxyproline residues, etc. And usually collagen is actually a triple helix and is a polymer formed of a repeating monomeric unit. If we take a look over the amino acid sequence of collagen, we will come across a repeating monomeric unit called glycine XY, and this tripeptide made up of glycine XY is actually polymerized to generate what is called a collagen triple helix. So what do X and Y amino acids correspond to this tripeptide of collagen? X and Y usually correspond to proline and 4-hydroxyproline in most of the cases. So the repeating sequence of tripeptide which is present in collagen is a monomeric amino acid sequence or unit, is made up of glycine, proline and 4-hydroxyproline residues. Now often in vertebrate collagens there are some unusual forms in which we have proline and 4-hydroxyproline replaced by a few by another amino acid, positively charged amino acid, that is arginine, or by a modified amino acid like 5-hydroxylysine, or simply by another positively charged amino acid called lysine residues. So instead of proline and 4-hydroxyproline, the X and Y positions are often occupied by lysine and 5-hydroxylysine, or are often occupied by lysine and histidine, that is arginine, likewise. So when lysine and 5-hydroxylysine are present instead of proline and 4-hydroxyproline, these amino acid residues are cross-linked by polymers and are cross-linked by special forms of covalent bonds, and when cross-linked they form special series of an unusual amino acid called dehydrohydroxylysine or leucine, which is dehydrohydroxylysine or leucine is a special form of tripeptide or unusual amino acid which happens to occur in collagen as when or due to the presence of the repeating tripeptide unit, that is glycine XY, where X and Y correspond to lysine, that is lysine and 5-hydroxylysine or arginine residues. So this is there are few other very special features of collagen which are strictly saying not conserved, but we have come across almost all those features of collagen which are conserved in all forms of vertebrate collagen. Now let us move on to another fibrous protein which is quite important. It is fibroin. It is the protein that is actually obtained from spider web. It is obtained from insects and is present in silk fibers. It's obtained from insects, fibroin of spider webs, etc. And it's made up of alternating units of beta plated sheets, that is beta conformation. And this fibroin, that is the protein present in silk, found in silk, obtained from spider webs, insects and spider webs, has a huge content of alanine and alternating glycine residues. So to summarize, we have altogether seen the structural aspects of three very important fibrous proteins. The first one which we discussed about was keratin. This next to that was collagen. We discussed about collagen and triple helix, and the third one which we discussed about is fibroin, that is the protein obtained from spider webs which is present in silk and is made up of repeating units of beta conformation and has a very high content of alanine and glycine residues. Now, other than fibrous proteins, we have a variety of globular proteins. Now, like serum albumin, which has 585 amino acid residues, as I said earlier, and consists of a single polypeptide chain. Then we have oxygen-binding hemoglobin and myoglobin proteins. We also have lysozyme, which is secreted and is present in animal tears, saliva, etc. Then we have ribonuclease C. It is a regulatory protein and a special form of globular protein, that is enzyme, that is being produced by pancreas and released into small intestine and seems to hydrolyze the covalent linkages found in ribonucleic acids present in food. Now, one more thing which I missed and which came to my fact at this moment, which while recalling, it just came to my mind. One more thing which we have missed is that collagen is derived a special form of protein which is very rich, which is very poor, which is not high protein, which has very poor proteinaceous content and which lacks almost all essential amino acid residues, and that which is derived from collagen